Whenever I'm ready. I'm ready now. Is everybody ready now? Okay, we're just gonna start. Okay. Hello, thanks for coming. I'm very happy to be here, given all the weird snow conditions at the airport. There's 3 screens and I've got a remote that highlights them, so you don't need to worry about where it is that I am looking. So you're interested about scientific communication, presumably for a variety of reasons. Maybe you want to know how to do it better. Maybe you have students who you would like to do better at it. Maybe you are very good at it and you were wondering how others do it. And in about an hour and a half, my hope is to make you more sensitive to some of these principles, some of these philosophies, more so than specific guidelines, because actually the more I do it, the more I think there are actually no guidelines. But there's a lot of different science figures out there, published or not published. Some of them are absolutely adorable and funny and possibly informative. If I'm doing heron at home, I want to know if I'm doing the right thing. But this graphic does not make its data clear. There are other graphics which are about exciting things. I bet you didn't have looking and copulating on your bingo card today. But while artistically presented, I feel like there's too much going on and I just gotta tell me what the information needs, although it does have redeeming qualities. On the other hand, there are some graphics which look full of great science, but lack any redeeming qualities whatsoever. And so the question is, how do we approach these ideas? How do we approach graphics like this, thinking about them so that maybe we don't make these kinds of graphics, but make graphics that are useful for the audience to propel their imagination and knowledge somewhat better. So there's 2 things that I want you to think about. What is it that you draw and what is it that you don't draw? You draw what is difficult to say. That is why we draw. We do not draw for the entertainment of the reader. We do not draw for our own satisfaction. We draw because things are hard to say. The converse of that is that we do not draw certain things. We do not draw what is easy to say, but difficult to draw. Let's take a little game from some graphics. What are these icons? Quick. Somebody drew these icons because presumably they thought they didn't want to say it. They wound up saying it anyway because it's difficult to understand. You don't know what that is, right? That means not practical. And this one, that means not comfortable. Wow. I know, and that's the graphic that it came from. Not practical, not comfortable. Let's play this game some more, okay? Instead, what you just do, you just say impractical, uncomfortable, programmable, unforgettable, that kind of thing. You just say it. Okay. Not all of you are in biology, but that doesn't matter. Even the people in biology are not going to get it. Perhaps you're thinking the last one, do not drink poison? You'd be very wrong, because actually means cell viability. Okay, this is the boss level. If you get this right, I will do whatever you whatever you want me to buy you at a bar. I will do it anyway, but I know nobody will get this. What does a mouse nailed to the wall mean? It means antidepressant like effect. Okay, so, and this is from this graphic. So what I've hoped to persuade you is that well-meaning smart scientists who go about their science in a rigorous way will do these kinds of things because they feel compelled to draw because they are drawing. Do not share that, do not go down that rabbit hole. So how do we fix one of those graphics? Don't worry, we'll come back to the fly sex later. So our first job is to make patterns clear and treat everything as data to the extent that's possible. So remember that mouse graphic where there was a lot of drug cocktails given to mice. How would we fix it? Well, we would think, what is the legend? The legend is a mouse because it's in mice. Dose A, dose B, some kind of a delivery, some kind of an effect. And then we would draw some axes and some drugs. And then we would connect the axes to create a cocktail. You see there's 4 cocktails, no problem. You couldn't possibly miss one. More or could be put in. You put in the effects and you're done. You don't need to draw anything else. In fact, you may not even need to draw the mouse because if you're talking about an experiment in a model animal, then it's a given that that's where it's happening. When you think about drawing, you're like, well, they told me not to use a table because tables aren't exciting, so I must draw. But tables are actually very exciting because they help you organize. And often the table is the right thing. So here is another graphic. Well-meaning scientist. I even know this person. who thought it was very important to show you exactly where the mouse needs to be poked with the medication, but only while having a drink. I don't understand. The point is, you can kind of see there's data here. Stuff goes up, stuff goes down. But what's the order of all the compounds? Why are we drawing the organs? We could draw the heart in a variety of ways. Which way do you think is best? Depends. Are you feeling dark? Are you feeling happy? Did you just fall in love? Or did you just, whatever? I don't think any of these are appropriate. I think that's what you need to say, because that tells you what organ are we talking about. So we set up a table. We have our organs. Now we can put them in some kind of an order that's meaningful. We drop in the up and down, upregulated, downregulated expression, and we put in some other stuff if we need to. Notice how we have no data for lungs in LPS ketone. That hole in that line is so clear. That's completely invisible in this original graphic. So this table can be expanded. Don't think of a table as this Excel spreadsheet, even though that's a kind of table. Tables can accommodate graphics. Tables can accommodate regions of data points that share some common theme, individual data points that are special. Look how this one stands out. Other data points where there's something going on, and so on. Impossible with the original graphic. This is what the scientists want. This is what I would want if I wanted to understand what is happening, if I wanted to have a systematic overview of things. And so when you draw, you should never think about what it is that you're drawing, but why. What is the function, and the form will follow. This is my firm belief. In other words, if you are telling me about something happening in cells and there's some channels and whatever it may be, what is the minimum amount of information I need to show you to make this message clear? Do I need to show you the cell shape? Almost certainly not. Do I need to show you the nucleus? It has nothing to do with anything. Do I need to make this subtle mistake, drawing the channels faded away in the background? Okay, I shouldn't walk right into that. No. To give you the impression that maybe channels start off as baby channels and grow into adult channels. If you're an alien who just landed, that's what you would think. That would be wrong. So I draw it this way. So there are two conditions. Here is the membrane. The membrane is not straight. Well, locally straight, but it's folded. It doesn't matter. Thermodynamics are not important in the message. There's some channels. Some of them stay behind. Some of them are extruded to the membrane. There is a cation that goes through, which I only need to label once. So far, the message is intact, and everything I'm drawing is about the message, some labels, and then I'm done. There's nothing that you can take away that will keep the message intact. Everything is necessary. You can pump up a little bit of the color if you like. You can make the arrows magenta to stand out, but those are subtle discretionary changes that are yours to make if you like. And you may have a few that you can make in a graphic, but most of the time you should take your cues from the information and from what nature is telling you. If it's a data graphic, what you want is to help people make good judgments. So if you show a data graphic, this is a simple data graphic, which is reasonably drawn. I don't know what I should be looking at. You're waiting for me to tell you what you should be looking at. Missed opportunity. So instead, if I want you to be looking at the second peak, I might highlight it. I might highlight a gray one, which is control, let's say. I might highlight a blue one, which is some kind of intervention. I might go further. I might think, well, you can't compute the areas on your head, so I might draw the areas or the heights, doesn't matter, in a bar chart. And I might say, 50%, there's some quantity above which it's important to notice that the peaks are higher or have a bigger area, so I will annotate that quantity. No other quantity is important. 50, which means below 50 is important, above 50 is important, but 60 and 70, there's no difference between them. And then I will think about what kind of question might you have in your head. I will give you the ratio of those peaks. And then as the start, I would start with that graphic, and then I would begin, talking or presenting, whatever, and you would have a lot to go on already. A lot of times you don't know where to look. You look at a complicated graphic and there's no zones of interest. And if you can inject that somehow, easier in a poster and a presentation, slightly harder in a journal because they have their own style, but this is important. So Journal of Neuroscience, I'm short because that's exactly what neuroscience looks like. I don't know where to look. I mean, I eventually figure it out, right? Yes, okay, but quickly, where does my eye go? It just goes outside. I check if it's still snowing. That's where it goes because I don't want to be looking at this. So even if you fix it and make it just perfect, you still are stuck, not really sure where to look first. But if I highlight the areas where there's a significant decrease, for example, the yellow says, look here. Oh, now you see there's a bunch of things to look at. All plots except one have some kind of a significant thing. You feel like, good, I can take them one at a time. This is very useful. And it can be a lot more subtle than this. This is pretty explicit. Look here, this yellow highlight, but sometimes it can be subtle. If you can emphasize trends, do so. It's very good for the reader. The reader, again, is busy, confused, possibly doesn't want to be here. And so they need some assistance, right? They need a little bit of a pep talk. So are you going to emphasize trends by torturing a visual metaphor? I don't think you should. I don't think you need to draw frustratingly two slightly different balances, telling me simply that things are out of balance. This makes me feel like I'm a 5-year-old. So what you, I think, should do is say, okay, here's the hypothesis. Here's the astrocyte, fine, releases some factor. In a normal case, there is a balance of inhibition and excitation. And importantly here, I've drawn these arrows up and down to tell you what the trend is in this particular case. And those kinds of arrows, this goes up, this goes down, are visual cues. Notice that I have kept those in a different color because I'm using the color as a theme, which we'll see again and again. This idea of the color is the hypothesis. The hypothesis is that there's a difference between normal and pre-symptomatic ALS. The idea is that in pre-symptomatic ALS, something goes up and down. That all is part of the theme. The rest is annotation. Sometimes it's really hard to find how to emphasize the trends because the quantities are complicated. Don't worry too much about this drawing. Yeah, I know not everybody here is in molecular biology or does vaccine work, doesn't matter. Because I'll walk you through the redesign and you will see, I think, what I mean. So here, there are these curves, and we're supposed to think about the trends in these quantities based on the shapes of these curves. I'm convinced that the shapes of these curves are actually wrong, but there's something else that is meant to be communicated. So the idea is, here's the background. If you stick somebody with a vaccine, they express proteins and you get antibody production, and so you have an immune response. Okay, there you go, that's the background. And then there's this poly A tail in the vaccine, and now we have the story. There are these enzymes, which are traditionally drawn as Pac-Man, whatever, that's fine. And they chop up this poly A, and what happens is that the mRNA and the protein levels fall. And see that half-life? I've marked the half-life. So now you're thinking, oh, that half-life quantity is important. I wonder what happens. So if I have a hypothesis that if I modify this poly A with some kind of a thing, doesn't matter what it is, some kind of a blue thing, now the enzyme can't get at it. And now the half-life increases, and I've drawn this as a same curve, just stretched. Here's the change in trend. Here's the increase of the half-life, explicit. Moreover, I've done even one more step, because depending on what kind of modification you make to the tail, you stand to gain potentially a further increase in half-life. Those two arrows essentially tell the story. And you can look at the arrows and go, oh, what's increasing? Half-life? Okay, I get it. Good luck with the original graphic. Good luck with it. I don't think it's possible. And so this idea of the story, what is the story? You know, sometimes the story, I don't think the story is the right word because the story has too many connotations of the street. I think a narrative is a good word, but whatever. It should have a beginning, a middle, and an end, and there should be some kind of a point to it. There should be some kind of a climax, if you will, or some kind of a moral, so to speak. Oh, I love this one. Graphical abstract drawn by student, no idea what it's about. It's about vestibular system, visual system, proprioceptive system goes into a brain, somebody's leaning over. And if you read the description of the project, what it is that they're proposing, none of the project description is in the visual abstract because this is not a well-drawn visual abstract because it completely misses the story. Here's some keywords that appeared in the project proposal. Greater risk aversion. Where do I see it? Don't see it. Reinforcement learning. Don't see it. Standing balance tasks. Okay, maybe I see the standing balance task. There he is. He's trying to balance. Risk-taking is essential. New movement strategies. Don't see it. What are they trying to do? Please help me figure it out. So here's what they're actually trying to do. So they got these patients, and then these patients are risk averse to certain positions because they can't keep balance. And so they go along this axis, and then they take some risks, but then they have an inversion, and then these are the standing balance tasks. And the idea is you want to take a patient so that they can now do more and then take risks that are greater, or what used to be risky doesn't feel risky anymore. And this is the quantity of change. This is the trend. The risk aversion has been pushed further down in the axis of standing balance tasks. Now, you're probably already getting a feeling for what's going on without me drawing anything, because I don't need to draw anything yet. Say, right, new moving strategies, there's some text, and now I drew the dudes. So there's a dude. The dude is like, I'm kind of risky. I don't want to do this, magenta. And over here, he's like, totally fine. I don't want to do this. But before, he didn't want to do it at all. Now he's slightly uncomfortable doing it. And then the idea is if you add in a little bit of a line here that shows you what axis his back is making, you emphasize the fact that it's about that imbalance. And then this little bit about what new movement strategies is, it kind of implies that to move your risk aversion over, you get him to do all sorts of different things, maybe move his arm, something like that. But that tells a good story. The colors show a progression of what is possible. And importantly, this arrow, I think, is absolutely key. So far, I think what you've seen is all the redesigns that I've shown you have a little bit more contrast going to them. There's a little bit more presence on the page, and that's very important because what I'm trying to do is I'm trying to map what is important to something that stands out to the eye. It's a lot of data, a lot of box plots. I don't know where to look. It's really kind of hard to see the changes because there's a lot of boxes. There's boxes and boxes and boxes around boxes. And you could feel like the whole thing is very flat and without any contrast whatsoever. So it's kind of like looking for Waldo, except it's worse than that because everyone is Waldo. Everyone could potentially be an important observation. They're hiding and you feel a little bit disappointed. So what would we do? We might do some things such as emphasize the position of the median, suppress all the y-axis ticks and only show the positions of the median. You can connect the medians with a dotted line, emphasize the trend. Even though that's not important, even though you can figure it out, that dotted line is an additional eye aid. You could go one step further and say, well, where females are values are bigger than male values, I will hint the background pink, let's say, and I will do blue for males. And I will put the ratio of the medians up there in the color. So now you can actually do even more computation and you can make comparisons. And then I will readjust it and reorder things so that at the top, I show you a legend. The legend is basically a graphic, which tells you what the colors mean and tells you what all of these tick marks are. I give you the age and BMI of the cohort because it kind of feels like that's what you should lead with. You should describe who you've got. And then I will put in some order that kind of makes sense to me, well, more than the original order, the panels. And it's a solid data figure that shows a lot of information. You still have to sort stuff out for sure, but it's a lot friendlier to the eye, and it is keeps in mind that we're not computers and we can't just image process the way machines do, that we need to find anchors on the page to focus our eyes. So I mentioned, are there rules? And I think that there are no rules. A lot of these guidelines that I may be proposing for one figure doesn't work for another. Don't let anybody tell you that you must follow this rule. Otherwise, things will go terribly for you. There's probably some exception to it. But there are principles that describe how your eye reacts to information and how it organizes information. And those are not negotiable. They merely describe how the eye reacts. So if I want to get your attention, I know there's a couple of ways of doing it. I can yell and shout, or I could make a pause. What a pause looks like on the page is like this. And pauses are very important. Essentially, space. Space is a very powerful tool that works in 2 dimensions. not just in one, like when we speak, because it's a timeline. So don't be that person. Things stand out when they are different from other things. Color, particularly when there is no color otherwise. A bit of foreshadowing about my feeling about color later. So if you put in a lot of color because you feel like nobody comes to your poster and you need to make it more attractive, this is what you've done. You've just taken the thing that's important and you've just hidden it. Not in a fun way either. So now to get more visual attention to the original thing, how am I going to emphasize it? I would do that. Just make it different enough. Notice how it stands out quite a lot, but it certainly does not stand out in the same way that the original individual red circle did. I now have to fight for your attention. Space is very important because it makes groups, which means it implies relationships when you group. So that if I bring things closer together, there's a relationship implied. Your mind automatically does this, and then you have to undo it when you realize maybe there is no relationship. Simultaneously, when things are far apart, you are implying some difference, or at least less of a relationship, which means boxes are not necessary. I know you probably love boxes. Everybody does. This is one of those therapy sessions that we can have later, where I tell you, you can drop the boxes. You're like, I cannot. You can't. Because it's about space. It's about the space. And you'll see me draw a lot of tinted boxes, but you will not find me dead drawing an outline box. Color, finally, is good for theme, but it's also a grouper. Again, things are seen as similar, therefore they will be grouped. If they are the same color, they will be seen as similar. Color is useful. Okay, so Gestalt principles are good to be aware of because there's some consequences of them that are almost always valuable to consider, such as this idea of inside and outside that you can achieve with this idea of a compartment. So, very nicely drawn atomic structures, some bases, some covalent bonds, which are the solid lines, some hydrogen bonds, which are the dotted lines. But notice how there's this frustrating aspect to this graphic where the covalent bonds make rings. Yes, I agree. And then now there's this ring made out of covalent and hydrogen bonds, and it feels like the whole thing is one structure. It doesn't feel like two structures being connected together by dotted lines. But if I fill in the rings of the covalent bonds, now you very strongly see two structures. That's Gestalt. That is this inside-outside thing. This is why tinting is so important. Light tinting, it's amazing. Just make sure that the projector can show it. You have a synaptic cleft. Well, you have trillions of them, I guess, so that's good. And do you get a strong feeling of inside and outside? You get a strong feeling that the cleft is outside and inside is the cell. Very weakly felt so. So what would I do? Other than cleaning up the text and all of that, fill in the cell, which should be a given, right? You'd probably already do that. But look how it strongly suggests this is outside and this is inside, very strongly. Finally, there's other applications of this idea of a compartment for pathways and cycles and things like this. If you fill them in, you have a very strong sense of a unity, particularly this yellow cycle and the gray cycle. Look how the boundary is so strongly implied. Here, it's very weak. The reason why this is useful is because it helps people remember, it helps people orient themselves among all of the complicated information, and it helps them just get on to the next slide without getting bogged down and trying to untangle the previous one. Because remember, nobody's just looking at this figure once a day, and that's the only figure they see. They probably look at hundreds of figures, and you want to make it as easy for them as possible. Yeah, so color. I think color should never be used unless it is meant to be a theme, that it means something, it stands for something, not just your personal preference, because I think science and understanding the world is complicated enough that as soon as you impose your personal preference on it, you run the risk of making it unnecessarily more complicated, not due to the data, but due to your own mood that day. So I would guard you against that mood. Here's a bunch of colors. Here's a bunch of tables. It looks kind of organized. It's a bit like a table, but what's the theme? I don't know. Proteins, there's a bunch of proteins listed here. Something happens. Protein extraction. I got to hunt for the word protein. PPI network, protein network. Okay, now the word is disappeared in an acronym. So what would I do? I would try to organize this in such a way that I would pick one color, let's say magenta, and I would encode the proteins in that color so that whenever you find a color, you think proteins, which means, oh, there's a bunch of names. Okay, they're magenta, fine, they're probably proteins. Oh, look, there's different protein. Oh, proteins are being extracted. There is colored shapes here. Why are they magenta? Because they're proteins. And here, I do some mass spec. I get different peaks, and these are different shapes, different proteins. And here I can maybe make some kind of a network out of them. So there are some little icons here to, make sure that people, when you take their clipboard away, they get very mad at me. So sometimes I feel like I need to put some of the clipboard back in so that they don't get angry. I don't think the clipboard is necessary, but fine. It tries to get out of the way. It does not interfere with your interpretation of how this magenta color flows through the graphic. That's a theme, and I think that's a really good use of color. All of these networks are different colors. This is completely unnecessary use of color. These are different colors too, these peaks. Where's the color here? Not necessary. A lot of times what people do is they use color to mean different. But you already know it's different because they have different shapes and they're already drawn as different entities. So you are now forced to use 5 colors, and there goes your Gestalt salience. There goes this idea that color can be used to stand out. And if you want to have more of a strong sense of an experimental protocol, you can make these panels, maybe even connect them. This is very discretionary. Connect them with lines, a little triangle saying this, then this, then this, although that's completely not necessary. However, I have used these triangles here elsewhere as well. So there's a bit of a visual vocabulary that's being set up, but that's not that important. In other words, something that replaces an arrow. Really, the only thing about an arrow that you need is the direction of the arrowhead. Strictly speaking, you don't need the arrow. So sometimes you can get away with just a triangle. Any questions up to now? Please interrupt me if you see something that upsets you or you disagree with, particularly if you disagree with it. If you agree with it, then we're happy. But if you want to tell me something, no? Okay. This experimental protocols, well, everybody has an experimental protocol. Well, everybody should have an experimental protocol if you're doing some kind of an experiment. Isn't it bad? But I have these very naive questions because I get a lot of graphics from fields of science that I am adjacent to, but I am not an expert in. And so I put myself in the noob position of asking very simple questions that maybe are very obvious to the scientists. But once they're asked, you realize, oh, they expose a weakness of the graphic. For example, this question is, how do I take a black chemical and a black dot, combine them to get a magenta dot? Where does the magenta come from? That's a very reasonable question. If magenta is a theme, where does it come from? Is it the particle? No, that's bluish. So that question doesn't get asked by the scientists because they're so busy, as they should be, on the science. And then they just pick colors and they're not deconstructing the material in a way that an audience, someone who's seen it for the first time might deconstruct it naturally. So what I would do is I'd linearize it. We start off with a particle which is hollow because there's nothing inside it. We take a magenta chemical and we put it inside, and now it's magenta. And then the lungs are magenta because we put it in the lung. They put magenta lungs here too, so that's great. But this idea of something is going into the particle is now intuitively encoded. And I don't think this is confusing. I think you get that. But what happens is that with something like this can extrapolate to a longer story in a poster where you can carry the theme and kind of propagate this idea of what the magenta is, because you know that magenta is now the strict black set. So here's an experimental protocol, and you feels like somebody has been drinking too much of the biorender juice, but also the arrows. Why are there's not even a horizontal or vertical arrows? Why are all the arrows the most frustratingly curved positions? Can I follow this? Is this a puzzle? Do I get a prize at the end? I feel like I should get a prize if I figure it out. Do I need to draw everything? Do I need to draw the spleen, or is that really a hot pepper? I don't know. So how would I approach it? Well, I would try to think, okay, the first thing that has to happen is there should be no curved arrows or arrows at an angle. So I'm going to try to place things that still are respect the process. You must respect the process, but that help you turn things into blocks. So, okay, here's the mouse. Here's some things going on. Here are some straight arrows. And then I would put in the labels. And I would be putting in the labels and drawing in the shapes kind of simultaneously, in the sense that I will not let one bully the other. I will not let the shapes crowd the area so that there's no room for labels, because labels are important. And I will not let the labels become disorganized on their own sense. And then let's say these things are important, so that yellow highlight. And then if these are objectives, for example, I would just do something like that. And that sets really nicely the context of what the priorities are. There's more room for turning things into blocks. Here is where I said that heart will come back. I feel like the mouse has a broken heart. It actually does. It has a heart attack. And I've drawn it up top. That's completely discretionary. It looks like a little playing card. My broken-hearted mouse beats you, or whatever. It looks like a tarot card, science tarot cards. So there's a fun idea. So you can redraw something, but it has to be done really, really, really subtly. And it can't be, oh my God, that's so witty, right? It can't be about you. It has to be about the reader, very much. Then what you do once you're finished, you could hide the layers in which you've placed, say, all the shapes, and you look at the arrows and you think about whether they look okay to you. Does anything look out of place? You know, I'm kind of now looking at this dotted line and I'm thinking, you see how this vertical arrow here is not in the middle of the gap between this dotted line and this arrow? And I'm thinking maybe that should be in the middle, just as that final way of organizing it. And then I look at the labels and I think, are the labels holding it together? They're kind of holding it together. Looks okay. It kind of does feel like there's spaces missing where there should be graphics, and that's the feeling you want to get. Because then when you see them together, things just feel really plated out really nicely. People love their zooms and crops. I don't mean to take your toys away from you. That's really important. I don't want to come across as someone who says that you shouldn't do all the things you're already doing. I think you should just do slightly less of some of the things that are hurting the reader. That's probably an important message. So here's a message that I think is hurting the reader. It's about surgery of a bone in the wrist. And notice that if frustratingly, this bone is drawn 3 times here, here, and here, each time a different way, at a different size. And it's a crop, right? I show you the hand over here. So I'm asking you to look at the bottom left of the page. I crop it so that I have a bigger space for it. I now ask your eye to move up, but I'm also asking your eye to move down because of this equipment. And then I'm asking your eye to move over to here. So the eye flow is very uncomfortable and not very natural. And the question is, how much space are you wasting for this crop? Can you not draw a crop? Can you draw everything once? I think you can. And I think because it's about the bone, I think the bone should really stand out and everything else should fade away. Yeah, the wrist has more bones than I've drawn. That's okay. You just put in however many you feel like are appropriate. But there's the main one, and it's broken. It really looks busted. It looks like ouch, and it's magenta, it's inflammation, it's the right color for it. And here's some equipment that we put in. And right here next to it are the ways in which we can get this wrong, because this screw gets put in that wrong order. And already you have a strong sense of what's going on. And the right things stand out, and I think the right things fall into the background. So the bone stands out, the screw stands out. That's why the screw has a white outline, is because if I put a black screw on a dark color like magenta, it would just get lost. White outlines are really great to emphasize the shape of something without having to draw a black outline, which can diminish the feeling of the color inside. Then I put in some important labels. So off target, too close, whatever. So I'm thinking, okay, these are the most important labels. I can't get those wrong. In other words, they have to kind of stand out. And then there are some supplementary labels, like the fact that these have optical markers and so on, that I hope kind of do not interfere with anything, because what I had was importantly, it was well laid out, and I don't want to add things that will make it worse. This is always the danger of drawing too much and too long, is that you have something that's good and then you add more to it and it's unnecessary, but you maybe are feeling insecure or timid, or you feel like, oh, I spent so much time on this, I really should add it in. It's an important, curious point that the reader will appreciate. And sometimes the reader doesn't appreciate because they've run out of patience before they can get to that point. If you want to make it stand out a little bit more, you could put a colored background. That is completely up to you. And so one of those guidelines that people might have told you is not to put saturated colors on top of saturated colors, because you get simultaneous contrast and it's uncomfortable to look at. So when you look at these bones, it's not that uncomfortable to look at, but you could see how red text on a blue background would be hard to read. But I have done that purposefully because the bone is broken. I've tried to give you this feeling of, oh, that hurts, that's uncomfortable. This kind of, that's called limbic resonance, this idea of communicating a deep emotional state, which I can't do because I can't connect my limbic system to yours, but I can try to do it through some experience that you are seeing on the outside, hoping that maybe it'll match. But it's not critical. It's just more of a, nice if it happens, but I hope you did get that feeling like that was a broken bone. That was the most important feeling.
My eyes are broken looking at it though.
Why?
It just looks like staring at a highlighter at 7 a.m. It's just too much to look at.
Right.
I got the idea of broken bones when I had the white background.
Yeah, exactly. Yeah, like this, right?
Mhm.
Yeah. So that's why I'm mentioning it. If you want to do this, you can, but that is your choice to do so.
Question there.
Yeah.
And this example, the optimal placement of the screw is the same color as the pad.
Oh, right.
Would that benefit from like maybe calling out that this is good?
Right. No, you're right. I have thought of it, and I think in the original graphic, the screw was a different color.
Okay.
So I thought about it.
Yeah.
And I thought about whether I should color. I don't think I should color the bone differently, because the bone is the bone. And if I now change the color of the bone, it isn't about the bone. It is about the position of the screw. It isn't even about the screw. It's about the angle that it makes. So if anything, what should be colored is some kind of a angle that measures this axis line with, say, the horizontal. So I would try to do it, but I don't. So maybe what could happen is maybe optimal could be bolded so that it stands a little bit more. This is that negotiation that happens inside my head when I redraw these things. And I think, okay, has the reader understood? Has the reader read optimal and therefore understood that it's optimal? Sometimes people say, well, I would like to see it visually encoded. I know, but sometimes it's not necessary to be visually encoded. Sometimes it is necessary to be visually encoded because optimal is difficult to understand. And so there's always that tension. Yeah. I tend to underdraw because I fix a lot of things that I think are overdrawn. So as the pendulum swings in this direction, right, I'm over in the underdrawn direction. And if I were to look at it and re-edit it, I would try to think of how to do it with a color, because then I would have to decide, okay, is off target worse than too close to bone surface? If I was ranking them, now am I implying more information? I would need to talk to the original person.
Thank you.
Yeah, no problem. So this graphic, remember this graphic? There's a lot going on in this graphic. And I don't think you have a chance to untangle it or find patterns in a reasonable amount of time, let's say 10 minutes. Maybe if you took it home and really took it apart, you would find things. But the idea is, why is it so complicated? Because there's a lot going on. So I think it needs to be taken apart and brought back together again. But what we will keep is the columns for the different disorders. So we'll draw some shapes that are kind of as simple as possible. These could be simpler. And we'll place them off to the side to kind of tell you what the relationships between them are, and point out the areas of each cell that are important and that we're going to look at. So far, you've kind of set up the list of actors. And then we'll have something to say about these individual parts, like the end feed, something happens, tight junction, whatever. There'll be some statements here that you can unpack for the reader. And now what we'll do is we'll have the columns for the disorders, and we'll take those vignettes and we'll place them where something happens, but we won't color them anymore because we've already blown through all our colors. They could just be gray because now we're going to be overlaying on top of that all the stuff that happens. And I'll be using arrows that are very salient to make it pretty clear, where all the changes in quantities are. I mean, it's still pretty complicated, but for example, over here, I try to make everything aligned to the extent possible. This is aligned, this label is aligned with this label, and so on. I'm not sure why I didn't draw anything here. Possibly I either missed it or this shouldn't be there. And then if there are places that are differentiated differently in males and females, you could annotate that with color. You might notice that some of that color is being reused. Maybe this is blue and this is blue. Maybe there could be some confusion. So I think here I changed the color slightly, but whether you change the colors or not, that's not that important. You would just pick something that maybe is a bit different. You could even annotate this, that the colors do not mean the same thing here. Maybe these colors are not necessary, although I feel like they're really nicely set up the theme for the astrocyte, the endothelial cell, and the parasite. But that would be something that you would think about, the choice of colors and whether it's working, and you would really, it could take time. It could take half an hour to figure out what a good set of colors would be for this. Not a nice looking set of colors, but one that is clear as introducing the cells and as introducing these areas. Yeah.
Do you have a particular color palette you like to use that takes into account color blindness?
Yeah, I usually pick magenta, blue, as you've seen me do, and orange. It usually works pretty well because magenta and orange stand apart from each other. So I try not to use red and green. And those are usually good. If I am uncertain because there's a lot of colors, I'll run some kind of a desktop simulator that shows me what a Deutera notebook sees, and I will take a look and go, okay, this is now confusing. Sometimes you cannot tell the color apart. People cannot tell colors apart, but they can still tell the luminance apart. So if something looks dark, something looks light, as long as they see the difference, that is good. So just keep in mind, color blindness is common, like maybe 4% of males. I think in a room of 8 men and 8 women, chances are even that someone is affected by color blindness. So your audience does not have to be a large audience for you to have someone who's affected, and running simulations. There's lots of software that does this, can be helpful. Just keep in mind that colorblind people can see neutrals as separate from color, just like you can tell grays separate from color. That channel is not disturbed. So this is a very difficult question to answer. You never know if you've answered it correctly because you never quite know exactly what your audience needs. Chances are the audience may show up to your presentation, to your poster, because they are so tired, they just need something fun to look at, like emojis on a pie chart, okay? Yep, sometimes that happens. You're tired, you're at a conference, and you're just like, oh, just show me something pretty. But that's probably not gonna be the case for everyone. So what is it that you pick? I think a clear explanation is good. If you can add a little bit of something to it, great, but sometimes adding that bit of something, like I mentioned, can destroy the explanation. So this is a very old book, but it has many good things to say about how to write. And I think many of them translate to drawing. So, right, what is ornate prose that's unwholesome and nauseating look like? To me, this is ornate. Is it nauseating? It's fun. I mean, I know Florian, he draws really well, and I could never draw flies like this ever, but I don't think I would want to in this case. So how would we fix it? What would we do? There's a lot of information here. There's a lot of neuroscience. There's a lot of stuff going on, and it's fun. It's about flies having sex. So if you look at his flies, they're made up of a lot of different parts. And I don't think, I look at the whiskers, or whatever the anatomy of the fly is. I'm sorry for any entomologists here. I just don't know all the fly parts, but there's a lot of different parts. And moreover, it's really hard to tell the males from the females apart because they're both kind of colorful, but I think their bodies are differently shaped, but can't tell the difference. So what I would do is I would just assign them colors, draw them in the same way, as simply as possible. There's probably a simpler way to do it, but I try to make them look slightly like flies. And so instead of, the female moving around and the male is moving around, and I'm trying to figure out, just stop moving around so I can figure out what's going on, I could do something like this, where the female will always be in the same position, and I will emphasize the movement through this arrow. So, and then some kind of activity through this concentric set of circles. So this idea of tapping, kind of like a vibration. And then he's singing, but we don't know how he's singing. So I think we need to say wing vibration. That's how he's singing, because he's not a good singer through his mouth, proboscis, or whatever the fly thing has in his head. Then there's the all-important licking, and then there's copulating, right? And if you want to add some personality to it, you can. But honestly, I think that that's probably already too much. But that's where I would add in the personality there. And so let's come back to this idea of the crops and zooms, and this idea of tell my eye, you must look here, then you must look here, you must look here, and the details about here are over here. So this idea, I just want the details to be provided to me where I am looking, if possible, if at all possible. Sometimes you can't get away from crops and zooms. Sometimes you just have to draw them, but sometimes you don't. So what I would do here is I would not repeat. I would ask myself, how many times do I have to repeat the pulse and the sign for you to get a strong sense that one follows the other, and then there's another pulse song coming? I think this is a good amount. And then I would highlight some parts. So what I've done is I kind of cropped and zoomed in place, but I'm just showing you the waveform, the underlying waveform. Let's say it's some kind of a triangle form. Here's a sine form. And then if I wanted to talk about carrier frequencies or something, or introduce a concept like interpulse interval, I would do that right here. And I would place those as a layer of annotation, and all the labels would be aligned and offset from the graphic. And those tinted windows kind of show tinted rectangles, kind of delineate the region of the graphic that is being discussed. Now that I look at this, I am thinking that possibly I should have made the entire circle inside blue, as opposed to extend the rectangle into the circle, if you see what I mean. I think that would have been better. Yeah, so there's always this idea that you haven't fixed it, you've simply improved it, and there may be other fixes and you don't know what those fixes are. But if you stare at something long enough, you'll get it to some reasonable place, and all of those other subtle fixes, version 2 next time around. But as you do this more and more, those fixes come to you faster because you've seen types of graphics like this before. So the last one for this circuits one. This is a lot going on, and it feels like the circuit is important to understand. There's a lot of detail here, but because the flies are taking up so much space, it's hard to get a feeling for the systematic patterns in the circuit. So what I would do is I would draw it like this, put them side by side. So, it explains this idea of he gets excited by his own wing vibration, and then there's exafference where she gets excited by his wing vibration. And then there are these tips on her antenna connected to some kind of anatomical structure that will feed into the circuit. And then there's a sensory part and a motor part, and then where they overlap, there's this dual tuning part. And then you would draw the circuit somehow in a way where all the arrows are as evenly spaced, have the right amount of length to accommodate the labels, very much like that experimental protocol we saw last time. But now the arrows have to be, are part of a circuit. There's loops and things like this. And now their ends, the ends of the arrows, have meaning in these kinds of circuit diagrams. And then we would draw the layers. And then this is the part that kind of feels like I am trying to change convention, but there's this convention of bidirectional inhibitory tracks, you know, kind of what they look like. And you could draw them like this, and that's how they were drawn before. But I kind of feel like that's a lot of detail that I would have to repeat over and over again. So I've set up a shortcut. Wherever you see this thick double line, that means bidirectional inhibited circuit. And this might be too much to accept if you're a neuroscientist and you're just not used to looking at, you know, two thick lines, and you want to see all those arrows. That might be too much. But I would argue that the busyness of the figure that would result would be a high price to pay, and your slight shift in interpreting what you see is a smaller price to pay. We have to pay a price looking at this figure either way. The question is, you know, which price do you want to pay? And remember how I said space makes groups, and if you put something close to something else, it might be interpreted as being part of that thing. So if you look here, the sensory label is gray, and it's next to this gray box, but it actually belongs to her and this magenta box. And so here what I've done is I have put the label on top of her and the label on top of him so that there's no ambiguity as to what sensory is labeling. You may have never thought that sensory might be labeling the gray box, but you might have because it was close to the gray box and it was also gray. You might also think, well, the box is gray and the flies are gray, so is there some kind of crosstalk in the signal of the color? And I would probably agree with you that there may be. And maybe the color of the fly or the box would need to be slightly different. Maybe if the box was slightly blue or something like that, then it would be very obvious that there's no interference with the gray flies. So any questions so far? Okay. So you probably have had to make a poster, and posters are kind of a default, one of the first times perhaps you get all the elements of your work together on one big artboard. And it's intimidating because you've got to worry about the flow of information. It's technically challenging to get everything together. And sometimes it's hard to know where to start. And if your lab has a template, you think that the template might help you. And it can kind of help you in a way of at least putting stuff on the page, but it doesn't necessarily help you tell the story because each story may require moving things around. So I want to show you a simple poster because posters can get really complicated and it's pretty exhausting to try to go through them all, particularly if you haven't seen them before. But this one is kind of small, but there's opportunities for change that you wouldn't consider because it's pretty straightforward poster. So it isn't like the poster is very, very confusing, but I will show you that by practicing on small examples, by fixing small examples and relatively uncomplicated examples, prepares you for the challenging cases where the case is no longer simple. So what is my problem with this poster? There's boxes, okay, that's fine. We'll get rid of all the boxes, lots of boxes. Remember, there's this rat, there's this colonoscopies, day 0, day 3, day 5. Later, there are some data plots. Day 0, day 3, day 5. So you're already thinking maybe there is some opportunity to connect the timeline of when the colonoscopies happen with the timeline that the data are connected. Why are they in different panels? Don't know. Maybe I'm thinking to combine these bar charts together. Maybe what I don't want to do is have a poster that reads like a paper, figure 1, figure 2. Remember, a poster is a prop, which for the purpose of presentation works with you as you explain it. And then later it's an exposition, maybe it's hanging on a wall somewhere and there's no one around. And now the person may be looking at the poster without your assistance. So posters have different lives. But if we can bring some of this annotation closer to the graphic, that would be wonderful. If we can remove some of the redundancy, such as what the stars mean in terms of p-value, that also would be wonderful. And maybe if we could do something with the conclusions, where the conclusions maybe aren't labeled conclusions, maybe they're labeled a little bit more actively, like what it is that I actually observed, and maybe they're not hidden at the bottom. There is a standard in science, which I think is very unhealthy for the communication of information, and that is to relegate to the most bottom part of the poster, ideally the bottom right corner where it's hard to see the most important things, which is your conclusions. Your conclusions are the most important things. The reason why I say that is because once I look at your conclusions and I go, whoa, those are good conclusions. Now I want to see how you got to those conclusions. I'm not going to just take those and run away with them and say amazing science. I want to be convinced that there's a process that was followed, but I would like to see the conclusions first. It's kind of the opposite of a story where I do want the punchline first. I want to see what the problem is. I want to see why the problem is hard to solve, how you solved it, or what do you observe during the solution, and then how you solve it. So when I redesign the posters, the first thing I do is I just move things over and try to get a feeling for how much space there is. This is kind of what maybe you would do if you were trying to break out of the template. So I will have a physical artboard, which is the size of the poster, 2 by 3 feet, 3 by 4 feet, whatever the poster size is. Usually 2 by 3 feet for this kind of thing is pretty reasonable. Maybe this is 2 by 4. I don't remember, but I'll move things around and get a feeling that this is how much text there is. This is the microscopy slides and so on. And there's, it's quite a lot of text, and I will try to think about what stories, how do the stories break down? What is important to highlight first? What is important to highlight second, and so on. And these are really nice, and I would like to have lots of room for those. So I will keep in mind that if I have enough room, I will want to make them as large as possible. So let's look at some of the graphics. And I picked this example because the graphics are really simple, which I think helps you understand them really quickly. But the ways in which they're fixed is kind of subtle. So for example, I feel like if you can combine axes where possible, or share axes again where possible, you should do so. So in this case, I've combined these by showing you the creeping fat mice as a group here, and the males and females as a group here. And then these are stars relative to healthy. I don't need this horizontal line that connects the pairs of boxes. I think if you have a lot of plots and those lines don't connect to everything, notice, right? These lines, there's all the pairs relative to healthy are shown. Maybe you could use them, but I like it like this because now all the stars are aligned. All the stars are, that sounds very poetic. All the stars are aligned, and you can compare them very quickly and see what kinds of significant relationships exist. Here's where the biggest, I think, opportunity for consolidating the plots is. So what you have here is you have these days, and there's colonoscopies during that days, and they have mice have a week, has a colonoscopy score, and then there's a different lesion area, I guess, of the tumors, depending on whether it's creeping fat or not. So I've drawn it kind of in keeping with how it's drawn over here, but I have made the contrast a little bit higher, so the data really stands out. The creeping fat is a worst case scenario, so I have made that magenta. And I've turned this box plot for the lesion area horizontal because what I want to do is I want to try to connect the colonoscopy score, CRF, no CRF, with the lesion area. So by placing it here, you can see how I'm kind of extending the theme of the magenta line over and the theme of the brown line. And a very powerful way to do that more would be this, would be to place the label for that data trace in between the plots so that this label is now annotating both this line and this plot like that. And so now if I drop my mouse and its journey through the experimental protocol, I see some opportunities for aligning things better. So this is a reasonable looking timeline, but this 035 days doesn't line up with this 035 days. So what I would do is I would try to make sure that my time axis, remember how I said it's a good idea to share axes? My time axis would be shared with all these plots. So here is the mouse. Here's when the colonoscopies happen. I've decided to put the colonoscopies on a separate track instead of drawing the colonoscopy dots on the timeline. Totally up to you. I wanted to show you that there's lots of room on this poster. So there's plenty of room to kind of untangle all the layers of information. And I think this works fine because it says, okay, here is the time length. And here are the outcomes of those observation schedules. I can, scan down 3 and see what happens with the weight change. I can look at the lesion area. And what I've done with all the text, I've moved the text closer to the plot so that if you wanted to know more about what you're seeing, it's right there, as opposed to a legend that you would need to unpack. And so the final poster looks like this. There's lots of room left, which is good, because I don't think you need to fill it. If there was more science to be put in there, great. But you do not feel like you need to pack the poster full of things. You don't feel like you need to make everything as big as possible so that the poster is packed. You need to make everything as big as possible to be comfortably read at a distance that somebody would be standing from the poster. And so I've put in headings like disease, model, and key observations. But I suspect that this idea of disease could even be more actively stated as, what is the challenge in Crohn's disease? Why are we studying it? The model, is there something that I could say as a headline that hints at what the model is? Think of a news headline. Think of something that the reader can already get by just looking at the heading. So if only the headings survive, then at least I have something. And then the key observations would be here. So this plot is, and this is the kind of the end of the text. It's under the key observations. And then the microscopy slides would be down below. All the call out lines would be straight. All of the texts would be aligned where possible, which is usually possible. Sometimes it's very tricky when the microscopy slides are on a black background. You could do some tricks with lines with a white outline to try to show the lines. So in the last little bit, I don't want you to get this feeling that I'm trying to suck the joy out of all the graphics. What I'm trying to do is separate out when the graphic should be informative and clear as quickly as possible, and put that in the graphic, and then take all the fun stuff out of the graphic and maybe put it somewhere else. Maybe on the title screen or something like this, where the audience gets the sense that you're emotionally invested, that you find your imagination is sparked by the work, and therefore maybe their imagination will be sparked as well. One of the worst things that can happen is that software cues you to do is click to add title. People just, I need a title, I have to add a title. So they may have a graphic, which is very compelling, but because they've clicked to add title, the text is interfering with my looking at the graphic. I'm not sure this is 100% correct, but I do agree with it in principle. And so always think about, I can't read and look at the same time. So I would just show this. And I, as the presenter, remember, this is a slide that there's a person standing there, go, water pollution is a challenge or something to that effect. And you would not need to read those words. I think this is better because now what happens is the muck goes off the screen. It stretches forever, which means there's muck over here. Whereas in the original one, it kind of ended, like, oh, but there's no muck here, so they must be okay. This guy just needs to swim over here. So this idea of open form, open meaning it goes beyond the frame, is very useful to imply something going on, something that's maybe infinite or very, very large. This is a great one. So the University of Sydney has this kind of burnt orange branding color, which I really like, from the poultry unit. And my question is, where are the chickens? Why is there such an uninteresting image of the poultry unit there? What could we put something there? It's our time to shine. It's time to show off the chickens. Let's do it. So why not just Google some chickens, whatever, doesn't matter. But pick something where there's something going on. He's here, it's open frame, he's just walked in. And he's got something to say to her, and she's not interested because she's eating. Whatever you think is going on, it doesn't matter. Everybody gets their own picture in their heads, and then you move on. What do you feel when you see this as a title slide? Are you curious to explore each of the shapes? There's like a little luggage and cheese, the web, little keys, that means security. What does it have to do with mouse? This is high detail, low effort. This is the day of AI, okay? Basically swamped with it. It does not even garner a second look. I just know this is somebody just did not want to think about it. But I was looking at the beautiful lines inside the bulb. See this, I like this line. This is the only thing that has imagination here. This bulb is not bad, but it's overdrawn. So what about drawing the mice in just one line like that? There you go. Picasso did this. I think this may either be Picasso or someone who drew them kind of in this style, but he has these animals that he drew with one stroke. They have a lot of personality. I think this is better. I don't think this gives you a feeling of low effort, high detail. I think this opens up ideas to the imagination and a bit of relief from detail, and then you get on with the detail in the mouse model. Okay, we're going to talk about snails. Is that what I put up there if I want to talk about snails? Is that the most exciting thing about a snail? If you've seen a snail, you know what a snail looks like. It's exactly like that. You've learned nothing about snails. Okay, maybe if you've never seen a snail, some people may have never seen a snail. This is the first time you're seeing a snail. That's for you. Congratulations, you now know what a snail looks like. But for other people, unless the snail is moving, no, it's not moving. So what I would do is I would pick something more different, something that I didn't know about snails. So snails make shells, okay? And those shells have funky colors. And here are the colors. There you go. And the last shell is turned a little bit just because. And that's it. And that's the title slide of a talk about snails. And it, I think, is a little bit more calming and is a little bit more imaginative. And maybe it makes you appreciate the snail in a slightly different way that the science wouldn't necessarily touch on the colors of the shells. Physics is exciting. Upper limits of energy non-conservation. Energy non-conservation sounds like somebody's breaking the rules. It's great. There's a picture in the back, but I can't quite make what it is because it's been hidden by this opaque white box. I don't know why somebody would do this. Somebody would want to shield you from this beautiful reactor detector. Look at these guys fixing all the elements in a pontoon. You could maybe put that horizon line in the middle for a balanced feeling. Maybe if you're feeling a little bit benthic, you would do this. I like this. If they fall in, it's toast, right? This is an uncomfortable image to look at. This is an image with some imbalance. You feel for these guys, like, are they going to be okay? Yeah, these guys will be fine. But these guys, oh, I hope they get paid enough for this. Okay.
It's energy non-conservation. It's already kind of makes you feel uncomfortable. So the uncomfortable.
Exactly. Yeah. Right. Exactly. So the title is so full of promise. And then you hide all the discomfort. Sometimes you got such great images that you want to put them all on the front page. You can't do that. You can only tell your best joke. So I think the one on the left is a better joke. And I would break with the law of thirds and put it right in the center, because if this was a crime scene and this was a blood spatter on the ground, and I was the forensic photographer, I would make sure that that is centered in the frame, because then I get the most of the context. Now, would I do that? Would I then just trample on the crime scene and put a title there and all of this other stuff? Notice how I have robbed this thing of mystery completely. So I would do something like this. I would put the title away from the thing as much as possible. Maybe now that I'm looking at it even further down. And make it really about that. And people would want to know what that is. People would be curious about this. People might want to look forward to the slide where that appears. So that was the last example. I hope this has been useful. I hope you've learned something. I will make the slides available. However they were to be distributed, I'm not sure. Please check with the organizers. And I guess we have a little bit more time left. I'd be happy to answer any questions. Did I say anything that will change your habits? Did I have made you reflect on those habits? Do you ever wonder where those habits came from? When you go to a therapist, they will say it's because your mom dropped you when you were 3 on your head. Or you had ice cream and you thought it was chocolate, but wound up being vanilla, and that's why you're disappointed for the rest of your life. But there is some reason why. Often the reasons because you pay them money, they have good stories. It feels like you're getting a good story here. You may have those habits simply because those are the habits you inherited from people in the field, that of papers that you're reading, people in your lab. The last person who made a poster, you just stripped out all the content and put stuff in a template. Basically, practical reason. There's no decision behind it. And I'm hoping that you will reflect more on the choices, on the fact that design is not a magical word. It's just a series of choices that you make intentionally, that you will draw things with greater intent. So when you place something over here, why did you put the red dot in the center? I have a reason. Is it the best reason in the world? No, but I have a reason. I've thought about it. If I say to you, why did you draw it here? And you say, I don't know, that's where there was room, then that probably needs a better reason. You're used to thinking before you speak, or at least thinking when you're writing. And when somebody says, why did you use this word? You have a good reason if you're speaking and somebody says, why did you use the word dog there? It's like, it's because I'm talking about a dog. Imagine if you said, I don't know. That would feel like somebody has something going on in their head that they need to get checked out, right? But with drawing, we're good with that. We're good accepting I don't know as an answer. And so I think once you start thinking about the reason why you draw and where you draw, just like you think about speaking, a lot of things fade away. A lot of habits that are just legacy decisions made by people we no longer know or are no longer around, or who knows what they are, no longer feel so heavy and things that you must follow. But it just takes time. It just takes time to make those decisions and think about them. And it may feel like it's a lot of work. It certainly is more work because thinking about something is more work than not thinking about it. However, you can wind up in a situation where by not thinking about it, you wind up futzing around on the poster, for example, for so long and moving things around. And because there isn't a structure or a cohesive set of principles that you're following, you could just be moving things forever and it'll never click for you because you don't know what the fitness function is. And so you actually wind up being stressed and using up a lot of time, more than if you had just started with a set of principles. So it will feel like you need to spend more time at the beginning, but very quickly you will get back. It'll be an investment and you will get a lot of that time back. That's my firm belief. That's the only way I can find a way to kind of sell you the time equation. But also good science requires time to communicate well, and it deserves it. If an experiment lasted for 2 years, 3 years, your PhD lasted for 4 or 5 years, an explanation deserves the time. You cannot, you should not feel that you need to shortcut that. You should refine it as much as possible. And those explanations will get better and better as the audience changes, as your own alignment with the science or the art or the communication changes too, will change. Yes, hey.
I had a question about thinking about like double encoding of information
Mhm.
graphics. So like the classic example would be like a scatter plot, and you have different groups that you encode not only with color, but also maybe shape.
Yeah,
And what are your thoughts? I mean,
Right.
you seem to have a very minimalist view.
I do, and that is, and I was going to say that sometimes double encoding is useful, but the example you laid out is one where I think it's almost never useful. Once you have picked a color for a line, you do not need a different shape for each trace. If you have a blue line and a magenta line and a gray line, they can all have circle data points. If the data points, and the data points can inherit their color if they are too far away from the plots, you know what I mean? But once the data points change shape, I don't think that that helps the reader, but because I think the reader can clearly see the difference between magenta and green, blue and gray. Where double encoding is useful is this helpful redundancy where you want to emphasize something. So you might color the text of a label next to something that already has color. The label is close to the thing you're labeling, therefore space makes groups, therefore it's unambiguously connected. I can share an example. I may have them somewhere. But if you color the text, sometimes it can drive home this point of a theme, even though it's really clear that the label belongs to the colored thing. And it's a question of how much accumulation of color you wish to achieve in one area, because that can help bring the theme to the reader a bit more because there's more color. So this idea of double encoding, I mean, I guess it means taking the same information and encoding it in multiple ways, that each channel is unambiguous and can stand on its own. With scatter plots, yeah, the shapes, I always pick circles. Circles are nice for shapes because they're overlap. The reason why a circle is like a magical shape is because an overlap of two circles never makes a circle unless it is identity, right? It's its own thing. Overlaps of triangles make triangles, overlap rectangles make rectangles. So a circle is good, particularly if you put a little white outline, a stroke around that circle. It helps distinguish one data point from the next. So it's one of those things where you should be aware that you're doing double encoding or you're being emphasizing things, and use your judgment, and you will never know. I try to be minimalist, and I'm trying to be less militantly minimalist, where before I'm like, no redundancy, never repeat yourself ever. So now I feel like, no, sometimes there's an opportunity for helping the reader view their eye, particularly with the balance of color, with the fact that you've thrown a bit more off to the side. So I think it's a good idea, but applied as gently to where you really need it. Somebody had a question about, yeah, hey.
Currently doing most of the graphic work, PowerPoint, in one chart, Excel. It's a.
Yeah. I mean, you should, are you asking for a friend or is that you? Oh, you're asking for a friend, yeah.
Kind of, like, I feel that so I've been doing graphic a lot, like with posters and my school and training in architecture department also on many of the visual things. But when it comes to like mentoring, say my own PhDs, it's like, they have 4 years. And not gonna ask them to do 4-year graphic work, right? So they probably only have like, I don't know, like
Sure.
a month or 2 months in their entire 4-year
Sure.
PhD to actually sit down and think, oh, I need to do this graphic good.
Yeah.
And to a decent level so that I can present it in conferences.
Yeah.
So are there like some sort of constructive I guess advice for those students, because that's actually one of the things.
Yeah. Tools, there's two things to think about. One is that the tool doesn't matter if you think about what it is that you want to draw. If you have a concept of what you want to draw, then any tool will do that can draw a circle and an arrow and a rectangle and fill them in. Simultaneously, to get to the point where you know what to draw, you need to have some experience drawing. And it's a bit of a with chicken and egg thing. So the tool can get in the way more than other tools. And there are some tools that can give you opportunities to do things that are very useful, such as those looking at the arrows and the text independently because they were in different layers and assessing their balance. PowerPoint doesn't have layers. So for Illustrator, Affinity Designer, there's Inkscape, there's a variety of choices do have layers. And a good tool is something that will ultimately make you faster at it, because what I'm proposing is drawing simply. And where the strength of the simple graphic comes from is their organization and spatial relationships. What that means is if you have tools that help you align and organize and snap and all of that, that's good. So I would say that the goal is not for these students to become master designers at no point, but they should be master explainers, at least, or beginner. They should get to a point where they can craft good explanations and with simple shapes. So while, yes, I think you can make do with PowerPoint, it can become very frustrating and you have very little room to grow within PowerPoint. And so you feel like a lot of the time you put into it is just you're burning the frustration, but you're getting nothing back from it. Don't get me wrong, I have sworn at all the other software packages quite a lot, but I do feel like at least there is I get something back once I figure out how to do something. So with, charts with Excel, okay, that's not necessarily bad. However, you need to be able to export those as vector art, EPS, PDF, whatever, bring them into some kind of a program where you can standardize text size, you can standardize line weight, you can standardize the balance between the data traces and the axis thickness. And that process it's a bit technical, and it does not take a long time to kind of wrap your head around. It's much simpler than the science itself, but it is very useful, so that when you send things to a journal and they all have their styles and they'll tell you something like, 0.5 point line width, 6-point minimum font, and someone has no idea what that means, then they may simply have their figures returned from the journal saying, this does not meet requirements, please reformat them, and be in a position where they don't even know how to do it. So, a PhD is a long time, and that is not to be completely spent doing design, but I think putting people in front of useful tools that don't get in the way, and then step by step at every opportunity that they do, change a habit, small habit change. After a while, they'll just get into it. And I also want to point out that in a lab meeting, it doesn't matter whether things are perfect. First of all, not perfect, but you could just show me the raw Excel plots because I know what I'm looking at, because we've been looking at the same process for the last 4 months. So I know what to expect. So that's fine. Don't worry about all the plots. But once there's an external audience, you start to worry about the plots more and more, to the point where the plots you worry the most about are in the grant proposal, because these are the people who have the least amount of time and the most amount of frustration, and they're looking for a scapegoat. And they're looking to just go, you know what, if they can't explain it to me, no. And those are the people who stand to change and influence your future. So that's why the clear explanation isn't simply to explain the science. I mean, as of course, I know you know this, but it's to make science possible by explaining it to the people with the money. And so that narrative, that flow becomes even that much more important. It's not easy, but it's a question of culture in the lab. If you set the culture to be, this is what we do. right? The student doesn't know that in some other lab, they don't do this. This is what we do, and this is why we do it. This is the reason. It's not my preference, and I'm not asking you to just use magenta, but, you know, and if I see purple, like I will spill my coffee on it. You would justify why you have these rules. You say, if you can come up with better rules, please let me know. And that it is part of the science. It's like keeping a good laboratory notebook. It's being detail oriented. All those things you do in the lab, you take for granted. If you're off by 0.1 degrees in your PCR, the whole thing can just go to shit. But there's no reason to believe that if you're off by 2 points on your chart, I live in a world where my world falls apart when it's misaligned. So it's it's culture, and so a lot of it falls to you, is to be that evangelist of good practice, good communication practices. But it's always about the good explanation. It's a clear message. Am I getting a strong sense of the process or the outcomes or the trends? That's what you should be looking for, not nitpicking people's, oh, slightly misaligned, because it's never that a figure can be improved because there's a slight misalignment on that one label. Generally, it's structurally unsound, and the explanation has been lost. Hey.
I'm gonna make 2 comments. I spent 20 years in industry before I switched to academia. And in industry, there's a lot of focus on simple things, because you have 10 people in a meeting, that's billable hours. So you don't have time to do a lot of complicated type of graphics. It has to be simple and to the point. 2 epiphanies occurred to me when I started my PhD process, and still I sometimes struggle with this. In engineering, a lot of our writing is procedures and CYA type stuff, and justifying things to a regulatory agency. In science, of course, the writing is the product. And when it came to figures and things like that, I remember some years ago muttering, I didn't know I was going to have to become a graphical artist. It's a good thing I like art. And doing the figures is still hugely important. As a matter of fact, with my students, I'm probably going to emphasize what you were saying, is that communicating both graphically and writing is going to be paramount if you want to continue along that academia track. My most recent paper, I had 12 co-authors, and I led it. And I had one person said, this paragraph is begging for a figure. So I put together a nice figure. Half of my co-authors said, I love this figure. It's great. It explains it. The other half is like, oh God, take it out. It complicates it. And I'm like, 6 in one hand, half goes in the other. I'm keeping it since I threw it. And I think it does add to the paper. It really adds to it, I would say.
Yeah, that's a good point, because you will get different responses from people.
Yeah.
And you have to weigh them carefully, because sometimes, even though they're not phrased well, they do have a point.
Mm-hmm.
I had this epiphany, a similar epiphany or a epiphany, when someone was pointing out that the poster I redesigned still had too much text in it. I took all the bullets out because I'm not a fan of bullets. And I said, but this is exactly the same amount of text as in the original poster. It's just that the bullets are gone. And then we had this conversation about what happens when people see bullets versus when they don't see bullets. And I came to the conclusion that I think the reason why people like bullets isn't because of bullets or that it's a list, but they expect that the sentence structure will be more compact, and it will not be as elaborate because people don't have to follow prepositions and phrases and all that. So there's going to be fundamentally less to read. That it'll somehow use less of their brain, I don't know. And then we got into this somewhat heated argument where I was incredulous of scientists wanting less to read and not more. Why would you be scared of text? Like, isn't that the whole point? And so that was important, is that someone who says too much text may actually be not looking at the volume of text, but be thinking, I don't want to read lengthy, tedious sentences or big paragraphs that are just a slog. I want it crisp and crunchy and sober. But if you already write this way, but somebody doesn't know this because they haven't read it, right? So people can have different opinions about what you do. You should think about what you've decided, stick with your guns, see where that goes. And after many iterations of that, have your moral compass that points in one direction. And if 6 out of 12, it's different with collaborators because It's a little bit more political. They're on the paper, whatever. They should have their say, but you should stand your ground and ask for reasons, and be ready to improve yourself or your view, of course. And understanding is that they should be ready to change their views. So this is hard. Everybody has, because the people come from different backgrounds, they have different amounts of training, and they have also different amounts of expectation of how good something can be. And there's this entire side of, if it looks too good, it must be fake. If it's too polished, you must be hiding something. There is this great piece in Nature written a while ago when they did rebranding, whether science should be ugly. Because if it isn't, then people get suspicious. All sorts of weird cultural things happen, and different fields have different takes on it. Younger fields are less susceptible to this, but older fields like this, you will take my chromatogram from my cold dead hands type of feeling, because I've been doing this for 7 years before you were even born, and when dinosaurs liked it. So what can you do? You have to figure out which hill to die on, so to speak, and those maybe is not the fight that you pick, but you should be thinking about it the most out of anybody around you, because then when you are, you have more foundation under those reasons, and the thinking is more cohesive, and it's always about what is good for the science and for the explanation and for the audience. It should always be about that. We're in the same boat. It's not about my ego. This should never be about, this is the way it should be because I say so. Of course not. It should be because looks like a better explanation. Maybe we should go with this one. I don't know. That kind of thing. It's very hard because especially when you're mentoring, because they say to me when I show them, it's like, oh yes, I like your figs, but my PI likes jitter plots. So I'm like, okay, do what your PI asks you to do, because I don't want to get in the way of that important relationship. Now, once you fledge the nest and go into the world, you could do what you like. So there's all sorts of people have all sorts of relationships with their supervisors. Some are very good, some are a little more tense, and so it becomes much more difficult than simply just changing the color of a data point. You know what I mean? Yeah.
I just want to comment that Martin is here tomorrow. Also, he's leading a workshop in Reiki from 4 to 6. And what that really means is a more open conversation about like what to do in maybe your figure or your science illustrations. It doesn't have data plots or anything.
Yeah, we're doing like a science art thing tomorrow, I guess. That's the theme, where if you want to take it to a little bit more of a emotional place, perhaps be creative with science materials or data or whatever. We'll talk about that. Yeah, so please come.
So this is part of Art in Silico, if you didn't know that, and Art in Silico's submission deadline. It could be just really beautiful plots, or it could be Canvas or multimedia or anything. The submission deadline is this Friday, and the show is next week. Thank you so much.
Thank you. Thank you.